Good morning and welcome to everybody around the world. Our topic for today is genetic causes of premature ovarian insufficiency. And I'm so delighted to have our illustrious speakers speaking today on this topic that is so important because when we have patients with premature ovarian insufficiency, in almost 80% of cases, we don't know why this has happened to them. And it's such a huge diagnosis for a young woman. So the agenda for today is that Professor Joop Laven will be speaking, and then there will be a recorded presentation by Dr. Hagerman because she lives in California and it's very early for her, but she will attend for the Q&A. I'll just talk and say that Bessens is our sponsor today, and we are very thankful for an unrestricted educational grant, but Bessens has had no role in the selection of topics or speakers. This has not been vetted or reviewed by them, and the decision for the speeches are made by a separate committee from the International Menopause Society. So with no further ado, I want to present Dr. Joop Laven. He is professor and head of the Division of Reproductive Endocrinology and Infertility at Erasmus University Medical Center in Rotterdam. He was initially trained and board certified in obstetrics and gynecology, and then in reproductive endocrinology and infertility. His research focuses on women's health, in particular reproductive endocrinology and menopause, as well as on infertility treatments and assisted reproductive techniques, and pregnancy outcome in infertile couples, especially women with PCOS. He has published over 350 peer-reviewed papers, reviews, chapters, and books, and has delivered over 300 lectures to international scientific audiences. He is also a visiting professor at the University of Shandong, China, the University of Adelaide in Australia, and he is a member of the International Guideline Group for PCOS, POI, and unexplained infertility within the Center of Research Excellence at Monash University, Melbourne, Australia. Thank you very much, Dr. Laven, for presenting this morning.
Thank you, Dr. Wolfman, for this nice introduction. It should be a short bio sketch, I would say, but I have to shorten it a little bit more, I guess. Anyway, it's afternoon for me, so good afternoon to the rest of the world. Without any further delay, I would like to disclose a few things to you, and especially the ones which are marked in red here are of importance for the content of my talk, which I'm going to give in the next 20 minutes or so. So what I would like to do with you is just look at what kind of single gene defects are known for POI. Then I would like to address some of the newer data from GWAS studies, which we did, and along with the Chinese people in Shandong. And then I would like to present a few animal models, and then end up with some general remarks on principles of ovarian aging, which is definitely different from, I guess, what you guys and I learned in medical school. So there is nearly one paper every month being published in which a new single gene defect is going to be presented, which is associated at least with premature ovarian insufficiency. And so what you actually see here is a kind of recent sum up in the OMIM database. I looked it up yesterday. And this is data which has been gathered till the end of 2022, in which all kinds of genetic defects are summarized on different chromosomes, which are associated with POI. And they are, if you would like to, let's say, categorize them, they are involved in disruption of oogenesis and meiosis, and I will come back to that later. But there are also genes which are involved in non-meiotic mechanisms, and they cause, in general, accelerated atresia or depletion of oocytes. And the other thing is that follicle dysfunction and or ovarian dysfunction is also very much involved, which makes sense, of course, because POI is, you have to look upon POI as a kind of accelerated loss of primordial follicles. Another way to do that is a gene ontology enrichment analysis, and this was done in this very recent paper, which just came out a few months ago, in which they looked at the 168 candidate genes which were identified with whole exome sequencing in POI patients. And what they tried to do is they would try to look at in which systems these genes were involved and what the role of these genes were. And the bigger the dot, the more reports are available of that specific gene into this specific area of interest. And so there are biological processes like reproductive structure development, sex differentiation, gonad development, meiotic cell cycles, but there are also quite a few which are involved in cell cycle processes. And they are having to do with condensed chromosomes, with the synaptonemal complex, which is primarily responsible for aligning genes during the first meiotic division. So it makes sense that these are very much in the cell cycle involved, and especially this process is happening in gametes. And since we are talking about oocytes, which are gametes as well. And then this latter group is a very important one. We discovered that already when we were looking at natural menopause, that the majority of genes which are involved in the age of menopause or associated with the age of menopause are DNA repair genes and DNA maintenance genes. And that's basically what is showing this picture as well. And so the number of genes is a little bit less here, but that has to do with the fact that they have recently being associated with premature ovarian insufficiency as well. Another way to look at that is what's done in this study, which was also published a few months ago. They looked at all kinds of different identified genes which were associated with POI, and these were all pathogenic genes. And they looked at also a few novel variants which they discovered using whole exome sequencing. And then they tried to categorize them into ones which were already detected and the ones which were undetected. So by using such a panel, you might detect quite a few which were previously not reported. Then they looked also at whether they were monoallelic or biallelic, or whether they had a kind of more heterogeneous and more multi-dependent kind of inheritance. They also tried to summarize what the individual impact of certain of these variants was on the total incidence of POI in this population. And then last but not least, they looked at whether they were involved in folliculogenesis and ovulation, in gonadogenesis, so the making of the gonad in the pre-embryo stage, and they looked also at oogenesis. And as you can see, the majority of these genetic variants are involved in oogenesis and folliculogenesis. And there's only very few ones which are, at least in this study, involved in, let's say, laying down the blueprint for the gonad. In the same study, they also looked at the known variants and new discovered variants, and whether they were also found, the same variants, in a population of controls. And what you see here, that's also posing a kind of problem into this field, that quite a few of those, like these ones here, they are also encountered in quite some cases in controls. And so it's questionable whether they are a pathogenic or likely pathogenic mutation. They might do that in these women especially, but if they also occur in a lot of controls without having their cause early menopause or very early menopause. Sorry. So that makes it a little bit less applicable, and it makes at least questionable whether these factors are the ones which are mainly involved as a single gene defect in POI, because they are also occurring in quite some frequencies in women without POI. Another interesting thing which they did in this study, they looked at whether there was a difference in women which had primary amenorrhea, so they never started to menstruate and had POI already before they entered menarche, or the ones which did that after they had menarche in a normal way. And so there are quite some differences in numbers here, but you can also see that there is quite a little bit of difference between the incidence of, for instance, the biallelic variants and the multi-heritage genes between women with secondary amenorrhea and primary amenorrhea. And they also looked at the rate of pathological and likely pathological variants in primary amenorrhea. And you can see that those are involved, those are the genes which are involved mainly in women with primary amenorrhea. And that's a different, let's say, set of genes, which is associated with these pathogenic variants in secondary amenorrhea, indicating that probably the pathophysiology and the genetic background is also different in women with secondary amenorrhea and primary amenorrhea. And to make a long story short, this is a very recently published, a very nice paper from the Italian group, which has quite a history as far as POI is concerned, and the genetics especially of POI. And they looked at all kinds of different gonadotropin independent germ cell line features. They also looked at the gonadotropin dependent follicles, and they were able to allocate a lot of these variants which they had found in previous studies, to allocate them according to all these different cell stages of a oocyte growing from a primordial follicle into a pre-ovulatory one. They also looked at some genetic variants which are happening outside the ovary actually, and they are summarized here, and they have to do with mitochondrial function. Keep that in mind because that's important, and I will come back to that later. And there are only a very few ones which are very much involved in laying down the blueprint of the ovary or the gonad in general, which are mentioned in here in this part of the slide. And then last but not least, there again, a lot of genes which are involved in meiosis and DNA repair systems. And when you look at all these different variants, it is tempting to look at whether there are several aspects which are very much focusing on theca cells, or whether there are defects happening mainly in granulosa cells or in oocytes. And this study, which is also recently published about 2 years ago, they looked at, they tried to categorize all these defects which they had encountered throughout the years, and try to allocate them to certain aspects of follicular genesis, like things which are critical for steroidogenesis, TGF-beta superfamily members, proteins critical for follicular genesis, transcription factors, and critical for downstream activation of follicular genesis. And I marked them here with the same colors as are summarized here. This is quite complex, but it's even more complex because there are a lot of factors which are indirectly having an influence on these systems. And so it seems tempting to look at whether we could have some gene panels which we could run in every patient with POI, and whether then we could see and whether we could diagnose what kind of mutation or gene defect they might have. But then this study has been published just a month ago, and this is a large study in which they looked at genes which were definitely associated or probably associated in yellow hair with POI, and genes which they identified after they did an extensive literature search. And they looked at whether these genes were inherited in an autosomal dominant or an autosomal recessive inheritance. And the interesting other thing what they did is they looked at the incidence of these variants in women with POI versus controls. And to make a long story short, if you walk down here along this line on the right hand side of each diagram, you can see the incidence first in women with POI, and then the incidence of the same variant in women without POI. And what they actually concluded is that the penetrance of these specific mutants was not that high in women with POI, and it was quite similar actually in a normal control population which had not yet had their menopause. So these gene panels might have a kind of disadvantage if you use them, because then you might at least get the impression that a certain variant is associated with POI. But then definitely you need to know the same, the frequency of the same variant in a normal population. And if that's similar, then it's highly questionable whether all these variants are the single variants causing POI. They are associated with POI, but not necessarily causally related to POI. So what about GWAS and next generation sequencing of POI? Has that been done quite a bit? Well, this is the first study, which was done by my friends in Shandong University in China, and they found one hit, which was genome-wide significant. And that was quite disappointing. There were quite a few hits in this area here from -4 till -6, which might have some role, and it's all about numbers, because this was done in a study in which there were only some 400 women with POI included in this GWAS. We did the same thing in 1,500 women with POI, and they had a definite definition of POI and a very strict one. And there again, we didn't find any hits which were above the level of genome-wide significance. We found, similarly as the Chinese, quite a few more hits in this area, which might be of interest. But then still, we are working on this, and it's definitely because we really need numbers. Just to put it a little bit in perspective for you, this is our study, which we published in Nature in 2021, late 2021, in which we identified 296 genetic variants in about 500,000 women in which we had data on natural age at menopause. So iatrogenic cases and other cases like surgical cases of menopause were excluded from this population. And the striking thing was, from the beginning onwards, when we did this in smaller studies, the group of genes which popped up being the most dominant ones determining menopause was DNA repair and maintenance. And there was about 20% of genes out of these 300 are involved in energy metabolism, and especially in energy metabolism in mitochondria. So if you look at what we anticipated way back when we started these studies in the beginning of the 2000s, we anticipated that we would find genetic variants which would postpone menopause for just a few days or weeks, or and other ones which were advancing menopause. But the effects were actually quite large, because if you look down here in this line in green indicated, the most lower one here postpones menopause for nearly a year, which is a huge effect for one single nucleotide variant. And there were also similar large effects seen, like 20 weeks advancement of menopause. So then you get your menopause 20 weeks earlier compared to the ones which were not bearing these variants. We also looked at whether there were differences between early menopause and POI cases. And generally, we did not see that much of differences in the early early POI cases, sorry. And there was also much overlap, which is shown here in this diagram in the left lower corner, and the effects on menopause and there was no that much of difference between cases which had early menopause or normal menopause and POI. What was interesting is that if you look at all kind of biological processes which are happening in the follicle, which is surrounded by granulosa cells and theca cells, that a lot of these things which we found, which were involved in mitochondrial DNA, but also in DNA repair pathways, and had roles in other, let's say, factors like ligands and receptors here, that the majority of the ones which we identified and our top hits, they were all involved in some processes which are very important for oocyte integrity and folliculogenesis as well, which gave us at least the impression that we are looking at hot leads. The other thing which we did in the Nature paper, we looked at genetic risk scores. And if you're in the upper tentile here and you have the highest risk score, indicating that you have the least effective variants, which are advancing your menopause, then you had about a 4.5, an odds ratio, which is posing 4.5 times more, let's say, often have menopause at age 45. And if we looked at in a different way at this genetic risk score, and we looked at the odds ratio for POI, that was approaching 5, which is comparable to bearing Fragile X pre-mutations, where the next speaker is going to address that. But indeed, so if you have a high genetic risk score, you have also high risk for developing POI when you have all the variants which are the least effective. This is an NGS study which has published recently, and they looked at different patients which are numbered down here up to 48, and they had also 10 patients in which they knew already the mutant which was in that patient. And the numbers in each bar are indicating the numbers of mutants which are in that same gene encountered. And the color designates whether it was it's involved in reproduction, for instance, like the green one here, or in cell cycle or signal transduction, as are the orange ones. And here again, to cut a long story short, you see that there are a lot of orange ones in here, which are very much involved in DNA repair and meiotic processes, which are derived in as being cell cycle specific. And a lot of these mutants bear more than one variant within that same variant which has been encountered. And they were able to use, when they used this gene panel, they were able to, let's say, identify 75% of cases which were previously not identified as being associated and probably also causatively related to POI. And so these NGS panels, they look very promising. Then a little bit of course, the proof of the pudding is in the eating, as we say. And we have to do that by just messing up DNA repair in animal models. We did that in 3 different animal models, and just focusing on this one, the ERCC1 mouse. That mouse is only investing energy in somatic maintenance. It's not investing in growth. They are very small and they die around 20 weeks of age, and they are completely naked most of the time. They are graying, they lose their hair. And a normal laboratory mouse, just to compare that, will live for 120 weeks. So they have a very much reduced lifespan. They are infertile and they are suffering from premature aging. And that is due to a defective DNA repair system. If you look at the, let's say, gonadal phenotype, it looks very much like the AMH knockouts, which we made in the late 90s, in which the ovary is depleted completely within 4 weeks, indicated by this high AMH level, which is a marker for the number of growing follicles in an ovary. And these levels are down to 0 and undetectable in the weeks thereafter. And if you look at the microscopic phenotype of the ovary, or the microscopic phenotype here, then you see that the ovary is already smaller at 4 weeks, and it's completely depleted of follicles after 15 or 16 weeks, which fits into what we see here based on AMH levels. So again, it is of no use if your soma is so defective and your soma is so much aging, so fast aging. It is no use to invest in a germ cell line, which is quite energy costly. And so then you deplete your ovary because you're not going to invest in that. What we did in a nature study, we introduced a CHEK2 knockout. CHEK2 is a gene which is very much involved in apoptosis. So it's an apoptosis inducing gene. And if you knock that out, you would expect that the number of antral follicles seen in these mice is higher. And that was the case after 1.5 months, but also after 13.5 months. So knocking out CHEK is indeed decreasing the number of apoptotic primordial follicles and leading to a higher number of antral follicles in the ovary. Introducing an extra CHEK1 gene, which is a gene which is inhibiting apoptosis, again, you see the same effect in more numbers at 1.5 months and also at 13.5 months. Afterwards, we superovulated these mice, and there again, if the CHEK2 knockout produces all the time more M2 follicles, M2 eggs, sorry, and the extra CHEK1 allele has a similar effect there. So modifying these genes is indeed also indicating that there is something happening in the ovary. This study is a study in which they looked at, they used, again, whole exome sequencing, and they identified a lot of single nucleotide variants which were associated with POI in this population. This is an Italian cohort, and there's also a smaller cohort, French-American cohort. And what they actually did in these studies, they looked at what the, let's say, the overlapping genes were between the Italian and the combined American-French cohort was. And then the ones which are overlapping here, they modified these genes, or some of them, and they did that in Drosophila, and so they looked at the size of the gonad, and this is the wild type. But you see, if you manipulate these genes, or if you knock them out, or you put an extra allele in there, you can see that you indeed modify the size of the gamete and probably also the function. But they did look at that into detail in this study. So there again, animal studies in which we try to, let's say, adjust the genetic function of these variants, they indeed show that there is a phenotype emerging which looks very much like premature ovarian insufficiency. So to sum up what I have said in the last 20 minutes or so, I guess, in the time when you were at 24 weeks gestational age, in your mom's tummy, you made about 7 million oocytes. At the time of birth, there were only 2 million left. And at the time you could really gonna use them at puberty here, you have about 400,000 to 600,000 left. And what you did before there, but also after that, if you have a lifestyle which is bad, and you smoke, for instance, and you use a lot of bad diets, what we like to call the Western diet, then you have more cumulative damage. And the way you repair that cumulative damage, that depends very much on your DNA repair and maintenance, that will allow you to extend your fertile lifespan beyond 40 years. And that implies that you also extend the age at which you enter menopause. So that's very much depending on how this system of DNA repair and maintenance is interacting with the amount or the load of cumulative damage together with the number you started with. And so it's a kind of intricate interplay between these three factors, which is very much causing the age at which you lose your fertility and the age at which you enter menopause. And that's basically completely run by aging. So it's not that women start aging after they enter menopause. They enter menopause because of aging. And the sooner you enter menopause, the sooner you are, or the faster you are aging. And that's also the relationship which makes sense that age at menopause is associated with the age at which you die. So to end up as my take-home messages, there are pathogenic and likely pathogenic variants known to be POI causative and also POI associated. The genotype-phenotype correlation indicates that genetic contribution was higher in cases with primary amenorrhea and secondary amenorrhea, and monogenic mutations are affecting folliculogenesis, ovulation, oogenesis, and gonadogenesis. POI is probably caused by multiple variants, other than in genes which are previously reported or currently evaluated in clinical panels. And genetic variants identified by GWAS overlap largely with those identified with menopause. So there is not that much of a difference yet detected. That could be numbers. It could also be that they share a similar genetic background anyway. And these genes are mainly involved in DNA maintenance and repair, and POI cases are likely oligogenic and polygenic in nature. So it's a kind of interplay between different genetic factors which causes the disease. Animal models indicate that a lot of the genes which we have identified are hot leads actually. And what I said, DNA damage is causing aging of the soma, and aging of the soma seems very much to be the predominant driver of loss of ovarian function. Thank you for your attention.
Thank you very much. Thank you very much, Dr. Laven. I didn't identify myself at the beginning of the webinar. My name is Dr. Wendy Wolfman. I'm professor of the Department of Obstetrics and Gynecology at the University of Toronto in Canada. And I run the Menopause and Premature Ovarian Insufficiency Clinic there. And also I'm a board member of the International Menopause Society and a co-chair of the educational committee for the International Menopause Society. So our next speaker is going to be presented on video as she is in California. We are really honored to have Dr. Randi Hagerman, a developmental and behavioral pediatrician, distinguished professor of pediatrics and medical director of the Mind Institute at UC Davis. She is internationally recognized as both a clinician and researcher in the field of neurodevelopmental disorders, including autism and Fragile X syndrome. She received her MD from Stanford, where she also did her residency. She then completed a fellowship in learning and disabilities and ambulatory peds at UC San Diego, then led developmental and behavioral pediatrics at the University of Colorado for 20 years. She co-founded the National Fragile X Foundation in 1984. In 2000, she joined the Mind Institute, and she carries out treatment trials for premutation carriers of Fragile X syndrome, and other neurodevelopmental disorders. She and her husband, Dr. Paul Hagerman, PhD, and her clinical team discovered the Fragile X-associated tremor ataxia syndrome, which is a neurodegenerative disorder associated with Fragile X premutation. She was also the senior author of the paper that first found a high rate of POI in carriers of the premutation. See the video now. Thank you.
Hello, my name is Randi Hagerman, and I'm a developmental and behavioral pediatrician at the University of California Davis Medical Center, specifically at the Mind Institute. And right in the corner here, I think you can see my pointer. This is a picture of the Mind Institute located in Sacramento, California. And here is our team. I'm going to be talking to you today about the Fragile X-associated primary ovarian insufficiency, or also called FAX-POI, This is actually the most common genetic cause of ovarian insufficiency, and it's caused by a mutation in the Fragile X gene. It's called the FMR1 gene, which stands for Fragile X associated ribonuclear protein gene 1. Now, there's two different mutations in the FMR1 gene. The pre-mutation is 55 to 200 CGG repeats on the front end of the gene, which is the promoter region. Now, everyone has this gene. Typically, it has less than 45 CGG repeats. It produces more normal levels of messenger RNA and normal levels of the Fragile X protein. However, another mutation in the gene is the full mutation, which means more than 200 CGG repeats. And this full mutation is typically associated with methylation or a methyl group on the DNA, throughout the DNA, which silences the gene. And this is the cause of Fragile X syndrome, because when the gene is silenced, there's little or no messenger RNA produced and no FMRP, and it's the lack of the FMRP that causes Fragile X syndrome, which is the most common inherited cause of intellectual disability and the most common single gene cause of autism. So anybody with autism or with intellectual disability needs a Fragile X DNA test. And it's about 1 in 4,000 who have Fragile X syndrome. But the pre-mutation, the 55 to 200 repeats, is much more common. And this causes just the opposite of Fragile X syndrome in that the gene is overactive. It produces too much messenger RNA, usually normal levels of the protein. And it is very common. It's about 1 in 200 females and about 1 in 400 males in the general population. So what I'm going to focus on is one of the pre-mutation disorders called FAX-POI, but there's also other pre-mutation disorders, the Fragile X-associated tremor ataxia syndrome and Fragile X-associated neuropsychiatric disorders, including depression, anxiety, ADHD, chronic fatigue, chronic pain, obsessive compulsive disorder, etc. So I'm going to touch on these, but focus mainly on FAX-POI because it's very important for people to test or do the Fragile X DNA test whenever there's ovarian insufficiency or menopause before age 41. Now, the expression of the Fragile X gene is demonstrated here. In the upper panel here, we have the messenger RNA level, and here we have the Fragile X protein level. And as you can see, after a gray zone that's 45 to 55 repeats, where the message tends to go up a little bit, and there's some evidence about a higher percentage of women that have FAX-POI in this gray zone, but it really, FAX-POI is mainly in this area here, which is between 55 and 200, or the pre-mutation region. And the messenger RNA goes way up, sometimes as high as 5 times normal. And then once the full mutation starts, if there's complete methylation, it drops down to nothing here if there's only partial methylation, or if it's unmethylated, and this is rare, but the messenger RNA can be higher. And the protein can begin to go down in the upper end of the pre-mutation range, as you see here. So you can have some features of Fragile X syndrome when the protein drops to about 30% of normal, but in the full mutation range, the features of Fragile X, such as long face, prominent ears, intellectual disability, hyperactivity, etc., manifests here. We're not going to talk very much about Fragile X syndrome, but instead about pre-mutation involvement. Now, there was early evidence of pre-mutation involvement. It started around 1987. The National Fragile X Foundation conferences include both family members and scientists. And I remember at a lunch table, there were about 4 women with the pre-mutation sitting with scientists, and the women were discussing how they all had early menopause at the table. And so we did a survey after that of about 105 women and divided them into those who were intellectually impaired, because at this time We weren't able, in the late 1980s, the gene was not yet discovered. It wasn't discovered until 1991. So we weren't able to tell who had the pre-mutation and who had the full mutation. But we divided them into those with intellectual impairment and those with normal IQ compared to controls. And 13% of the normal IQ carrier women had early menopause versus none of the intellectually impaired and about 5% of controls. So this showed an increase. It wasn't quite significant then, but subsequent studies demonstrated that early menopause is seen in about 16 to 20% of pre-mutation women. Also, there was early evidence that depression was more common in the not intellectually impaired carriers at that time in the late 1980s. And so subsequent studies have demonstrated that the prevalence of early menopause is associated with a CGG repeat. And those with very low repeats or those with very high repeats usually did not have early menopause, but the high frequency of early menopause was between 70 or 80 CGG repeats up to about 120 or 130 repeats. So it was very much related to the CGG repetitive sequence. And so this is demonstrated in another way. The FAX-POI risk for carriers to develop early menopause relates to the CGG repeats. And as you can see here, the age of menopause is related to the CGG repetitive sequence. So it seems like the mechanism of early menopause and some of the other premutation disorders is related to the toxicity of these CGG repeat numbers. And the greatest toxicity is kind of in the mid-range of CGG repeats. In the upper range, above 120 repeats, remember that FMRP levels drop, and that may have something to do with why the higher number is protected. And remember, in the lower range, the CGG repeat number is not as high. So another factor that is important for reproductive risks to have a child with Fragile X syndrome are the AGG anchors. Now, remember, in general, a carrier woman has one X with a normal FMR1 gene and the other X with the premutation. So overall, there's about a 50% risk that that X with the premutation will be passed on to the next generation. Most of the time, when the repeat number is 90 or greater, it'll go to a full mutation. So when there's 90 or more CGG repeats, that number will go to a full mutation in her offspring when that mutated X is passed on. But another factor that can change that risk are the AGG anchors. Now, about every 10 CGG repeats has an AGG anchor. And some individuals have no anchors, some individuals have one anchor, and other individuals have two anchors. So the more anchors you have, the lower the risk of going to a full mutation. And this graph looks at the risk of having a child with the full mutation. As I mentioned, after about 90 repeats, unrelated basically to how many anchors you have, the risk is about 100% when that X is passed on. However, if you have no anchors, that risk goes up depending on how high the CGG repeat is. So at about, say, 75 CGG repeats in the mother, the risk it'll go to a full mutation is about 85%. However, if she has one anchor here, the risk drops to about 78%. But at 75 repeats, if she has two anchors, the risk is about 30%. So again, it can only pass to a full mutation when passed on through a female. If a carrier male has children, he has only one X, and that X is passed on to all of his daughters, and it stays as a premutation. So a carrier male will pass on the premutation to all of his daughters. It only expands to a full mutation when it's passed on by a woman. If a woman has a full mutation, then that X, when it passes on, will stay as a full mutation. But again, depending on how many anchors, her risk will decrease. So with 2 anchors, the risk goes down significantly. So it's important for genetic counselors to have this risk chart available and to know how many AGG anchors a carrier woman has. Here's another way to present this. This is expansion of a premutation to a full mutation. Depends again on the repeat size and the AGG anchors. And this just shows 2 anchors are in the light color here. Again, as you can see, no anchors has the highest risk. 1 anchor has slightly lower risk, and 2 anchors have way lower risk. This just shows the lowest risk here with a low CGG repeat number. So it's important for you to know that there are several Fragile X disorders. Fragile X syndrome is the full mutation first described actually in a 1949 pedigree, Martin Bell pedigree. The first X-linked pedigree was later shown to have the Fragile X mutation. And then Herb Lubbs discovered the fragile site on the X chromosome in 1969, and the gene, the FMR1 gene, was sequenced in 1991. But again, there are several premutation disorders, including FAXTAS, FAXPOI, which we just talked about, but also FAXAN, which is Fragile X-associated neuropsychiatric disorders, and that includes a variety of neuropsychiatric or behavior problems, including ADHD, autism, ASD, anxiety disorders. This can start in childhood, but many of the psychiatric problems, particularly insomnia and depression and anxiety, are seen in adulthood. So many of the mothers you see that have early menopause will also have depression or anxiety, and that occurs in about 50%. So the most severe problem of the free mutation is FAXTAS. Let me just show you a video of what it looks like. It's tremor and ataxia. This is the intention tremor of a woman here with FAXTAS. We first described this condition in 2001. And so the finger to nose test will show the intention tremor. Usually they don't have a resting tremor, but occasionally they do. So this is a Parkinsonian symptom, and it interferes with handwriting and pouring and eating when it is severe. This can be associated with cognitive decline and white matter disease in the brain. As you can see here, this is involvement of the middle cerebellar peduncles. It's like 2 headlights looking out at you. There can be brain atrophy, but there's also inclusions, FAXTAS inclusions in the neurons and the astrocytes, but not in the oligodendrocytes. This just shows these inclusions occur throughout the brain. This is just a picture of these inclusions in the neurons and glial cells in the amygdala. Males have more more dementia and more white matter disease and more severe involvement than females, because females are protected by the second X. These inclusions can occur throughout the central nervous system, but throughout the body. Inclusions can also be, for instance, in the testicles, in male premutation carriers, and they are in the Leydig cells that produce testosterone. So premutation males, before they develop FAXTAS in their 60s, may have erectile dysfunction and testosterone deficiency. This can include inclusions throughout the ganglia of the intestines and throughout the spinal cord and the thyroid, kidney, even heart. And in fact, cardiac arrhythmias are related to some of the toxicity of these inclusions. here's the first gentleman that demonstrated inclusions. These are, again, in neurons and astrocytes, and they stain in a certain way. They're tau negative, but H&E positive. This just shows some of the white matter disease, periventricular white matter disease, middle cerebellar peduncles, and it also can involve the splenium of the corpus callosum. Now, why is there pre-mutation disorders? This is because the excess RNA causes toxicity, often related to sequestration of proteins like SAM68, DROSHA, DGCR8, that are important for neuronal function and cell function. And when they're sequestered by the excess message, it causes toxicity. There's some evidence of RAND translation, which means repeat associated non-AUG start site translation, which can produce a protein that's called FMR poly G that may be toxic to the central nervous system. There's also mitochondrial dysfunction in pre-mutation carriers, calcium dysregulation, and all of this leads to white matter disease. There can be environmental toxins, for instance, those that are alcoholic or addicted to opioids or go through chemotherapy, or smokers can have more rapid progression and earlier onset of FACS-TAS. There's also iron deposition in the brain, and those with chronic traumatic encephalopathy like boxers can also have earlier onset of FACS-TAS. So the spectrum of pre-mutation involvement can occur in childhood, and we see about 10% of male carriers can have ASD. ADHD is very common. Usually, we don't see intellectual disability in pre-mutation carriers, but again, we see psychiatric problems like anxiety, stress, depression, POI, or other types of endocrine dysfunction like hypothyroidism is very common, and about 5% can develop other autoimmune disease like lupus or MS, and neurological problems such as neuropathy, migraine headaches, chronic pain or fatigue, restless leg syndrome, insomnia, memory problems, foggy thinking, even high blood pressure, and as I mentioned, erectile dysfunction can develop before the onset of FACS-TAS. And most of the time, when individuals present with tremor or ataxia, they have executive function deficits, memory problems, and about 50% of the males eventually develop dementia. So the toxicity of the pre-mutation can cause enhanced cell death. Neurons die earlier in culture, and that's why they're sensitive to like environmental toxins or even second genetic hits. So if we see a person with the pre-mutation and intellectual disability, we usually do whole genome sequencing to look for second genetic hits that would cause intellectual disability. So treatment of pre-mutation carriers include many things. We treat the neuropsychiatric problems, usually with SSRIs. Exercise stimulates neurogenesis. We like to reduce oxidative stress with supplements, antioxidants in the diet. We like to alleviate any factors that lead to CNS deterioration like high blood pressure or hypothyroidism, avoid toxins if possible. And we think that isofluranes from general anesthesia can be toxic. And so if they have general anesthesia, we try to avoid isofluranes used by the anesthesiologist. And also cognitive stimulation is very important. I have some more slides about psychiatric problems, which can occur actually in about 50% of adults. Anxiety and untreated can lead to shrinkage of the hippocampus, whether it's with or without FACS-TAS, as you see here. And psychiatric problems can become worse over time. These are 2 episodes of testing separated by 2 or 3 years. So anxiety, mood disorders can increase over time in pre-mutation carriers. We talked about how it can start in childhood. And many people thought that the psychiatric problems were just related to raising a child with Fragile X syndrome, and that certainly can worsen stress. But this Marshfield study validated FAXAN, where more than 20,000 people in the Marshfield Healthcare Network were tested for the pre-mutation, and neither the physician or the patient was told about the pre-mutation. And what was found is that psychiatric problems like agoraphobia, social phobia, panic disorder, chronic pain problems came out as significantly different from the general population in both males and females. Females had more of these problems here, but other related psychiatric problems, mood problems, major depressive disorder were significantly increased in males. And this can occur even when you don't have a child with Fragile X syndrome. This just shows there can be a clustering of conditions. So these are individuals with FAXPOI, and this is just a heat map showing how much more common these problems listed here. And FAXPOI, along with some mental health problems such as anxiety, occurs here, but there are some that have a more complex profile. And for instance, with chronic muscle pain, chronic fatigue, fibromyalgia can occur here. Also, sleep problems, even without FAXPOI, can be associated with other disorders to a higher rate. But then there's a group of premutation women who don't have any of these problems. So again, the FAXAN problems occur in about 50%. So smoking and having a high BMI or obesity, depression or anxiety can increase the odds ratio for these other problems. So anxiety can increase the odds ratio for depression, obsessive compulsive disorder, chronic fatigue, social phobia, fibromyalgia, chronic muscle pain, sleep problems, ADHD, etc. And these problems, again, get worse as you get older. So now we know that FAXTAS problems start before, actually after the age of 50 in the 50s or 60s, but sometimes executive function deficits can occur earlier in the lifespan. For instance, those children or young adults with ADHD often have executive function problems, and these just show how common executive function deficits or selective attention or inhibition problems occur across the decades of life. And we think that the same kind of RNA toxicity can occur again throughout life. So in childhood, it's mainly maybe learning disabilities, FAXAN symptoms like ADHD. FAXPOI starts in young adulthood, FAXAN continues, and in later adulthood, sometimes the anxiety, stress, depression can get worse, along with hypertension, migraines. And in the elderly, 50 and older, average age of FAXTAS is about age 62. That's when more significant neurodegeneration can occur. We hope to have better treatments. You can treat symptomatically the tremor with primidone and the psychiatric problems with SSRIs, but we're really looking at something that could reverse some of the RNA toxicity. Allopregnanolone can do it in cell culture, but we hope to start an oral allopregnanolone study funded by the Department of Defense next year. This year, we're working with biomarkers to follow these problems over time. And then Anavex is another treatment. It's a sigma-1 agonist that works at the mitochondria and the endoplasmic reticulum that can reduce mitochondrial dysfunction, help with protein misfolding, modulating calcium, reducing oxidative stress, reducing inflammation, and enabling neuroprotection. So in the future, there should be better treatments, and some of these treatments will probably be helpful even for FAXPOI. So I hope I have stimulated you about how important it is to test for the premutation. All women with any type of ovarian dysfunction or infertility, when the cause is not known, should have Fragile X DNA testing, and that's very easy to do. Send to most molecular laboratories at a university or private labs like LabCorp or some of the other private labs in the UK. If they have a premutation, then you need to think of all of these other problems that can occur with the premutation, and think about investigating the whole family tree for other premutation and full mutation problems. I want to thank my collaborators. Here's our Fragile X team here enjoying a great meal. And if you have any questions, I'm happy to answer your questions. Thank you very much.
Some of the participants would like to pose some. Here's one. What is the availability of genetic testing for POI in the clinic setting? I think it is available in many universities. I'm not sure in the various countries around the world. So you'd have to find out. But I think if anybody has POI, they should have at least chromosomes and a test for Fragile X. So I think if you have that sort of person that you are seeing, you should either try and find out where you can get that test done or send them to a reproductive endocrinologist who can. So if there are no further questions, I'd like to really thank Dr. Laven and Dr. Hagerman for 2 excellent lectures. I learned a lot and I hope you did too. And I'd just like to give a shout out for the 19th International World Congress on Menopause in Melbourne between the 19th to 22nd of October 2024. In Canada, we have a Canadian Menopause Society meeting in November in Vancouver between the 9th to the 11th, 23. So I thank you very much again for attending today, and I thank you to our lectures and also Besson for sponsoring this event. Thank you.